Gadus morhua, also known as the Atlantic cod, is a benthopelagic fish which is vital in fisheries in Europe and North America. Cod stocks have experienced collapses due to overfishing, especially northern cod, which collapsed in the 1990s due to the stock being depleted at a rate faster than it could be replenished. Currently, cod stocks are still under threat throughout the world, with many stocks still being overfished and below the target level biomass. As a result of this, it is important to have methods of estimating the population of Atlantic cod to ensure that they can be sustainably exploited as a resource. To improve fishery management, there is an increasing need to understand the current state of populations. One method of estimating current cod populations is using catch data. For example, when there is a major decline in catch rates, it can indicate the state of a current population. If the size of catches decrease, it can indicate that a fish species is being overfished. Many of the world's commercial fish stocks are in decline, raising concerns about food security and ecosystem functioning. Stock assessments for cod can often be inaccurate. For example, a former stock assessment from 2008 forecasted that stocks would rebound. However, they were once again on the verge of collapse by 2012. A study by K.R. Peterson assessed the reasons for inaccuracy in predictions of major cod stocks. The study found that catch rates of Gadus morhua in several regions have been misreported, presenting a key limit in using this method for population estimates. The study states that fisheries regulation can impose strong incentives on fishermen to underreport landings, with regulatory agencies often being unable to prevent illegal landings or landings that go over established fishing quotas. For ICES stocks at present, it is suspected that official reports of catches are strongly biased by misreporting, and that research vessel information may be less biased, though with a larger error of variance. Virtual population analysis is a cohort modeling technique commonly used in fisheries science for reconstructing historical fish numbers at age using information on death of individuals each year. This death is usually partitioned into catch by fisheries and natural mortality. Analytic methods based on virtual population analysis mostly rely on the assumption that precise catch at age information is available. One such method that has been used for population analysis on cod stocks is the Lauric Shepard method. This method accepts the catch at age data to be exact, which is a key limit in its accuracy, as this data is rarely exactly correct. After this, the method then adapts the resulting range of possible population biomass estimates to fit auxiliary data, depicting trends in the biomass. Auxiliary data is often commercial and survey catch rates, which, as previously established, can often be inaccurate due to misreporting, further impacting the accuracy of the Lauric Shepard method. A paper by Kell et al. Evaluating this method found that this method can produce wide margins of error. This study showed that several methods used for virtual population analysis can produce inaccurate results. This figure shows that for many stocks, stimulated trajectories do not reach the implied equilibrium level during the stimulated period, such as the North Sea cod, where the stock collapses despite predictions, the Western Baltic cod, where the stock oscillates in the vicinity of the expected point of the curve, and the Eastern Baltic cod, where the stock is underestimated by the management procedure. An advantage of this method, however, is that it calculates mortality rates, allowing for comparison in fishing mortality rates over the years. This method can also help calculate shrinkage to the mean fishing mortality, which is useful when trends in fishing mortality are variable over the course of several years. The main virtue of the Lauric Shepard method is its simplicity and ease of understanding. It can help guide decision-making when trends in fishing mortality are not certain. As such, this method has been used extensively in the ICES arena, especially for stocks of North Sea cod, haddock, and Western Baltic cod. Sustainable exploitation of marine populations is a challenging task, relying on information about their current and past abundance. Fisheries-related data can be scarce and unreliable, making it unsustainable for quantitative modeling. An alternative method to using fisheries-related data consists in estimating the effective population size, a concept founded in population genetics. Effective population size is the number of individuals in a population who contribute offspring to the next generation. This method is undertaken using eDNA, which can help identify the presence and abundance of fish species. Increasingly, attention is paid to the genetic state of marine populations, with numerous studies being published on genetic diversity, genetic population connectivity, and genetic population size. For example, data from the Web of Science shows that between 2000 and 2017, the annual number of publications estimating effective population size of marine species has increased sixfold. The pros of this method is that it is useful for evaluating the genetic future of marine populations, be they harvested or not, as reductions in effective population size are positively correlated with reductions in population viability. A key disadvantage in using this method for marine populations is that obtaining large numbers of samples, usually in the form of scales and tissues, can be difficult, and genotyping costs can limit sample sizes. As a consequence, most sample sizes are under 1,000 individuals, as shown in this figure. In North Sea cod, this can lead to less than 1% of the population being sampled, such as in a study by PLoS ONE et al. However, an advantage of this study is that it allowed research to explore the genetic composition of two different cod populations in the North Sea and the Bornholm Basin. This method allows genetic comparisons of the same species across different populations and within the same population over a period of time. For example, the previously mentioned study by PLoS ONE found significant genetic changes in the Bornholm Basin population. However, the small sample size used in this study is a key drawback, and a larger sample size would yield more precise results. Several studies using this method have small sample sizes. For example, a study undertaken by Zahivo-Stotsky et al. In 2016 attempted to estimate the effective population size for cod in the Barents Sea using a sample size of only 43 samples. Despite its small population size, however, this study showed that a landlocked population of Atlantic cod in Kilton Lake had distinct morphological features and a faster growth rate and age of maturation than sea cod due to being in a different environment. So, despite the limitations in small sample sizes, these studies can still be useful in determining the dynamics of a population, even if they have limits in quantifying population size. For short-term predictions used in stock assessment, early and precise estimates of recruitment are essential. In heavily fished stocks, like Western Baltic cod, where the age structure is dominated by a few age groups with a high contribution of first-time spawners, such predictions are of basic importance. As is obvious from time series of spawning stock biomass and recruitment, the Western Baltic cod has fluctuated considerably during the past 40 years. Moreover, recruitment success also changed significantly, indicating either that spawning stock biomass is not an adequate measure of reproductive success, or that environmental conditions and potential species interactions influence recruitment. To make any predictions about recruitment, it is necessary to consider all the steps of the life cycle, such as stock origin of the recruits, distribution and migration of parents, stock structure, including mean age and mean size of spawning fish, spawning areas and time, the number spawned, stock composition, maturation, and fecundity, including the destination and drift of early life stages, and factors limiting the survival of larvae, such as environmental factors, predation, and human influence. Identification of spawning grounds is often based on ichthyoplankton surveys, where the densest areas of egg distribution are assumed to be where the adults spawn. There are two major problems with this approach. First, egg distribution does not necessarily reflect the spawning areas, because eggs' buoyancy may cause cod eggs to sink to greater depths if the salinity is low. In this case, quantitative sampling is difficult, because it is impossible to sample close to the seabed near the greatest concentration of eggs. Second, in a hydrographically highly variable environment, such as the Western Baltic Sea, with high wind speeds, the distribution of eggs and early larvae are influenced by prevailing current and may easily, within a few days, be entrained far from their spawning origin. Identification of spawning areas from the results of plankton surveys may lead to incorrect conclusions. Validation by comparing the distribution of running adults is necessary. In this study, errors in abundance estimates were highest at high wind speeds and when the organisms showed a patchy distribution. And the results suggest that wind forcing might serve as a rough approximation for the reliability of historic abundance estimates. Warmer water leads to earlier spawning and a shorter spawning season than cooler water. Another factor that may influence spawning time is salinity, in that suboptimal salinity leads to a cessation of spawning time in laboratory experiments, and apparently also in the wild. Studies of adults with running gonads that support these interannual variations unfortunately do not exist. The results of this sampling method differ based on the time of year in which it is undertaken, due to changes in rates of spawning. Studies undertaken in Mecklenburg Bay, such as this study by Bleil et al. In 2000, showed limited spawning activity. However, it is possible that these results were because these studies were undertaken in times of a year with limited spawning. It is especially hard to work out rates of spawning activities in areas where spawning is undertaken once a year. A useful tool for studying the impact of early life stage entrainment is hydrodynamic modeling. However, there are considerable interannual differences in drift destination, depending on wind speed, direction, and timing in relation to spawning. In years with strong westerly winds early in the year, eggs and larvae may drift from the Kiel Bay/Danish belts as far as the Arkona and Bornholm basins. As many as 70% of all eggs and larvae could be advected into the Arkona basin, and 30% as far as the Bornholm basin. In years with low or moderate wind during spawning, larvae seem to be retained in the spawning area. Also, situations with variable wind directions tend to retain the larvae in the spawning area. The major shortcoming of the application is that seeding of the model with drifters, i.e. eggs and larvae, and drifter behavior, i.e. vertical migration, is not adapted to interannual variability in spatial stock dynamics, mainly through the lack of appropriate data.